I am Troy Handlovic, and this is my project titled The Quantification of Estrogenic Compounds by SPE, Solid Phase Extraction, Gas Chromatography, and Mass Spectrometry in New Jersey Natural and Wastewater Sources under the mentorship of Dr. Salierno and Dr. Elshaer. For a quick introduction, estrogenic hormones are in a class of chemicals known as endocrine disrupting compounds. And these EDCs interact with the endocrine system and cause undesired results at extremely low concentrations, since they're active at concentrations similar to circulating hormones at the parts per trillion level. And since they interact with the endocrine system, they disrupt reproductive behavior, which makes their danger even more profound since it can be transgenerational for both vertebrates and invertebrates. The synthetic estrogen of interest for us was 17-alpha phenylestradiol, EE2 for short. EE2 is used with progesterone by over 100 million people as the oral contraceptive commonly known as the birth control pill. So when an individual consumes the birth control pill, around 40% of the ingested EE2 is excreted by the body. Those excretions make their way through wastewater, eventually to the wastewater treatment plant, where it's treated, but due to inadequate wastewater treatment, a fraction of all excretions, pharmaceuticals and excretions, make their way out to the river. So the goal of our project was to find out if EE2 and a few natural estrogens were making it out into the river through this wastewater treatment effluent, and how much was there to see if it was dangerous. So studies like this have been done before over the years, but were mostly done in Europe and Asia. The amount of estrogens in the water in the United States is grossly unknown. So this was like a local project, so we could compare the concentrations to known toxicological assays and see if our local New Jersey aquatic organisms were in any danger. So the last point I have for the introduction is it's estimated that around 30,000 kilograms of natural estrogens and 700 kilograms of EE2 are excreted by the human population yearly. So it's a significant amount. The four estrogens that we were interested in look at were called estrone, based estradiol, 17-alpha phenylestradiol, that EE2, and estriol. Three out of the four, E1, E2, and E3 are natural, so it can be excreted by animals or humans. And EE2 is that synthetic estrogen that we were interested in. So we had to design a method to separate these estrogens from one another and to concentrate them so that we could quantify them individually. I have below some chemical and physical characteristics of the species. And what's important to note is that they are relatively large and have relatively high boiling points for GC analysis. So we'll come back to how we prepared them to make them more suitable for the GC-MS. Before we could prepare our samples to be injected into the GC-MS, we had to concentrate the samples since we knew that we would expect estrogens to be in the parts per trillion, the nanogram per liter concentration if they were present in New Jersey's wastewater and natural water from previous findings that were done in those studies overseas. So in order to do this, we started by concentrating 2 liters of samples from either the river near wastewater treatment plants or the wastewater treatment effluent itself. We passed that through a solid phase extraction column where we see the sorbent on the left and the estrogens were adsorbed onto the column. After those 2 liters were pumped through the column, we dried it and then reconstituted the sample in 1 milliliter. So this allowed us to do a 2000 to 1 concentration and to get the estrogens at a high enough concentration to analyze using single quadruple GC-MS. And so as I mentioned before, the estrogens were large for GC-MS and had high boiling points. So to make them more suitable, after we concentrate them through the SPE, we reacted them to create the TMS derivatives. So instead of having a hydrogen on the hydroxyl groups, we replace the hydrogen with trimethylsilyl groups, and this allows the compounds to be more readily separated and detected through the GC-MS system. The slide I have here is the only point of the qualitative parts of the project that I'll talk about, since I do want to talk about quantitation. But what this shows is the black on the top is the mass spectrums that we recorded, and the blue on the bottom is the NIST webbook equivalent for all 4 estrogens in their TMS derivative form. So this confirmed that for 1, we separated the estrogen successfully, and for 2, we synthesized their TMS derivatives. When we do quantitative GC-MS analysis, we use 9-methylanthracene as the internal standard for internal calibrations. That's shown here. This was chosen since it has a fused ring structure as well and is readily available. And while doing GC-MS in the quantitative form, we instruct the mass spec to analyze in SIM mode, so selected ion monitoring, where we analyze for only these 10 compounds, these 10 ions, I mean, sorry. And by analyzing only these 10 ions, we're allowed to make more exact and more rapid measurements, which allows the mass spec to be much more powerful quantitative detector. But the problem with SIM mode is you no longer have that full mass spectrum fragmentation pattern that we saw before in those head to tail plots. So we had to come up with an alternative way to identify the species from these unknown samples. And to do that, we followed direction from the official Journal of the European Communities, where they designed this 4-part identification system. And the first part of that was determining the retention times of all of the compounds. So as you can see here, the standard deviation in the retention times is low, and they elute very similarly each time over these 18 replicates. So we added plus or minus 0.1 minutes to the retention time to create these time windows. And that was the first part of our SIM identification procedure, where we said if estrone was in the wastewater, it would have to elute at this time window. And the second part would be it would have to have this presence of this quantitative ion that we knew was unique to the species from computational studies that were done before. It would have to have the presence of the conformational ion. And if you divided the signal from the quantitative ion by the signal from the conformational ion, you would need to see an ion ratio that's within 10% of 1.37. So if all 4 of those steps were achieved, we could be confident to say that estrone was in the wastewater and move on to quantitation. What I'm showing here is the MS component tables that you see after using that system that we described, where the top is the quantitative channel and the bottom is the conformational channel. If you were to divide the signal in terms of area of the peak in counts per minute of the quantitative channel on the top by the signal of the conformational channel on the bottom, you would get that ion ratio, which is unique to each species. And if you were to sum up these two peaks, you would get the SIM chromatogram that we see provided by the software shown here. And what this shows us is that all 4 estrogens were successfully separated. And we see the time windows on the top, and the time windows allow us to only look for the 2 ions of interest within each window to make more rapid measurements. After we designed the method that had them separated.
Sorry, I apologize, but we are really running very late. So if you could address the conclusions, it would be good because we have
Sure.
to give everybody a chance. Again, I apologize.
No problem. This is the most important table. So unfortunately, due to the pandemic, we haven't been able to get samples from the wastewater treatment plant. But what this shows here is that when we took 2-liter samples and spiked them at 100 parts per trillion, we have an average recovery after extracting and concentrating, separating, and quantifying of 70.5%, which is above the EPA minimum recovery for analytical GC-MS work at 50.5%. So this showed that our method was valid and that it allowed us to do some preliminary sampling. We sampled from these 2 wastewater treatment plants where we sampled in the river next to the wastewater treatment plant and the effluent as well. And that was right before the pandemic. So unfortunately, we were able to detect the estrogens, but we haven't been able to quantify them yet since we haven't been permitted to get samples yet. So in terms of further studies, we hope to get out to these sites in the summer now that our method is designed and valid and quantify those estrogens so that we could compare them to the toxicological assays. In terms of acknowledgements, I'll just acknowledge my mentor, Dr. Elshaer and Dr. Salierno, and then I'll take any questions.